So hello students, as we know reading is the first step towards learning. So read with me. Today we are going to study this chapter unit 3 cell structure and functions. Biology is the study of living organisms and the detailed description of their form and appearance only brought out their diversity. It is the cell theory that emphasizes the unity underlying this diversity of forms. It means the cellular organization of all life forms. A description of cell structure and cell growth by division is given by the chapters comprising this unit. Cell theory also created a sense of mystery around living phenomena. It means physiological and behavioral processes. This mystery was a requirement of integrity of cellular organization for living phenomena to be demonstrated or observed. In studying and understanding the physiological and behavioral processes, one can take physico-chemical approach to use cell-free systems to investigate. This approach enables us to describe the various processes in molecular terms. The approach is established by analysis of living tissues for elements and compounds. It will tell us what types of organic compound are present in living organism. In the next stage, one can ask questions: what are these compounds doing inside a cell, and in what way they carry out gross physiological processes like digestion, excretion, memory, defense, recognition, etc. In other words, we answer the question: what is the molecular basis of all physiological processes? It can also explain the abnormal processes that occur during any diseased condition. This physico-chemical approach to study and understand living organisms is called reductionist biology. The concept or technique of physics and chemistry are applied to understand biology. In chapter 9 of this unit, a brief description of biomolecules is provided. G.N. Ramchandran, an outstanding figure in the field of protein structure, was the founder of Madras School of Conformational Analysis of Biopolymers. His discovery of the triple helical structure of collagen, published in Nature in 1954, and his analysis allowed information of proteins through the use of Ramchandran plot rank among the most outstanding contribution in structural biology. He was born on 8 October 1920 in a smaller town not far from Cochin on the southwestern coast of India. His father was a professor of mathematics at a local college and thus had considerable influence in shaping Ramchandran's interest in mathematics. After completing his school years, Ramchandran graduated in 1914 as the top-ranking student in BSc Honours Physics course of the University of Madras. He received PhD from Cambridge University in 1949. While at Cambridge, Ramchandran met Linus Pauling and was deeply influenced by his publication of models of alpha helix and beta sheet structure that directed his attention of solving the structure of collagen. He passed away at the age of 78 on April 7, 2001. Chapter 8: Cell, the unit of life. When you look around, you see both living and non-living things. You must have wondered and asked yourself, what is it that makes an organism living, or what is it that an inanimate thing does not have, which our living thing has? The answer to this is presence of the basic unit of life, the cell, in all living organisms. All organisms are composed of cells. Some are composed of single cell and called unicellular organisms, while others, like us, composed of many cells, are called multicellular organisms. What is a cell? Unicellular organisms are capable of independent existence and performing the essential function of life. Anything less than complete structure of a cell does not ensure independent living. Hence, a cell is the fundamental structure and functional unit of all living organisms. Antony van Leeuwenhoek first saw and described a live cell. Robert Brown later discovered the nucleus. The invention of microscope and its improvement leading to the electron microscope revealed all the structural details of the cell. Cell theory. In 1838, Matthias Leiden and a German botanist and examined a large number of plants and observed that all plants are composed of different kinds of cells which form the tissues of plant. At about the same time, Theodor Swan, a German zoologist, studied different types of animal cells and reported that cells has thin outer layer which is today known as plasma membrane. He also concluded, based on his studies on plant tissues, that the presence of cell wall is a unique character of plant cells. On the basis of this, Swan proposed the hypothesis that the bodies of animals and plants are composed of cells and products of cells. Slayton and Swan together formulated the cell theory. This, however, did not explain as to how new cells were formed. Rudolf Virchow first explained that cell divided and new cells are formed from pre-existing cells omnis cellula acelluli. He modified the hypothesis of Slayton and Swan to give the cell theory a final shape. Cell theory as I understand today is all living organisms are composed of cells and product of cells. All cells arise from pre-existing cells. An overview of cells. You have earlier observed the cells in an onion peel or on human cheek cells under the microscope. Let us recollect their structure. The onion cell, which is just typical plant cell, has a distinct cell wall and as its outer boundary, and just within it is the cell membrane. The cells of the human cheek have an outer membrane as the delimiting structure of the cell. Inside each cell is a dense membrane bound structure called nucleus. This nucleus contains chromosomes, which in turn contain the genetic material DNA. Cells that have membrane bound nuclei are called eukaryotic, whereas cells that lack a membrane bound nucleus are prokaryotic. In both prokaryotic and eukaryotic cells, a semi-fluid matrix called cytoplasm occupies the volume of the cell. The cytoplasm is the main arena of cellular activities in both plant and animal cells. Various chemical reactions occur in it to keep the cell in the living state. Besides the nucleus, the eukaryotic cells have other membrane-bound distinct structures called organelles like the endoplasmic reticulum, Golgi complex, lysosome, mitochondria, microbodies, and vacuoles. In the prokaryotic cell lack such membrane-bound organelles. Ribosomes are non-membrane-bound organelles found in all cells, both eukaryotic as well as prokaryotes. Within the cell, ribosomes are found not only cytoplasm but also within two organelles, chloroplasts and mitochondria, and on rough ER. And because it is found, it is called rough ER. Animal cell contain another non-membrane bound organelle called centrosome, which help in cell division. Cell differ greatly in size, shape, and activities. For example, mycoplasma, the smallest cell, are only 0.3 micrometer in length, while bacteria could be 3 to 5 micrometer. The largest isolated single cell is the egg of an ostrich. Among multicellular organisms, human red blood cells are about 7 micrometer in diameter. Nerve cells are some of the longest cells also vary greatly in their shape. They may be disc-like, polygonal, columnar, cuboid, thread-like, or even irregular. The shape of the cell may vary with the function they perform. Prokaryotic cells. The prokaryotic cells are represented by bacteria, blue green algae, mycoplasma, and PPLO. Pleuropneumonia-like organisms. They are generally small and multiply more rapidly than the eukaryotic cell. They may vary greatly in shape and size. The four basic steps of bacteria are Bacillus, Coccus, Vibrio, and Spirillum. The organization of prokaryotic cell is fundamentally similar even though prokaryotes exhibit a wide variety of shapes and functions. All prokaryotes have a cell wall surrounding the cell membrane except in mycoplasma. The semi-fluid matrix filling the cell is the cytoplasm. There is no well-defined nucleus. The genetic material is basically naked, not even enveloped by a nuclear membrane. In addition to the genomic DNA, many bacteria have small circular DNA outside the genomic DNA. These smaller DNA are called plasmids. The plasmid DNA confirms certain unique phenotypic characters to such bacteria. One such character is resistant to antibiotics. In higher classes, you will learn that this plasmid DNA is used to monitor bacterial transformation with foreign DNA. Nuclear membrane is found in eukaryotes. No organelles like the ones in eukaryotes are found in prokaryotic cells except for ribosomes. Prokaryotes have something unique in the form of inclusions. A specialized differentiated form of cell membrane called mesosome is the characteristic of prokaryotes. They are essentially infolding of cell membrane. Cell envelope and its modification. Then we have to read ribosomes and inclusion bodies. Then eukaryotic cells. Then cell membrane and cell wall. Here are the organelles, mitochondria, plastids, ribosomes, cilia and flagella, nucleus, and microbodies. And most prokaryotic cells, particularly the bacterial cells, have a chemically complex cell envelope. The cell envelope consists of a tightly bound three-layered structure. It means the outermost glycocalyx followed by cell wall and then plasma membrane. Although each layer of the envelope performs distinct functions, they act together as a single protective unit. Bacteria can be classified into two groups on the basis of differences in cell envelopes and the manner in which they respond to the staining procedure developed by Gram. Those that take up the gram stain are gram positive and the other that do not are called gram negative bacteria. Glycocalyx differs in composition and thickness among different bacteria. It could be loose sheath called slime layer. In some foil it may be thick and tough called capsule. The cell wall determines the shape of the cell and provides a strong structural support to prevent the bacterium from bursting or collapsing. The plasma membrane is selectively permeable in nature and interacts with the outside world. This membrane is similar structure to that of the eukaryotes. A special membranous structure is mesosome, which is formed by the extension of plasma membrane into the cell. These interactions are in form of vesicles, tubules, and lamella. They help in the cell wall formation, DNA replication, distribution to daughter cell. They also help in respiration, secretion processes to increase the surface area of plasma membrane and enzymatic content. In some prokaryote like cyanobacteria, there are other membranous extension into the cytoplasm called chromatophores, which contain pigments. Bacterial cells may be motile or non-motile. If motile, they have thin filamentous extension from their cell wall called flagella. Bacteria so range in the number and arrangement of flagella. Bacterial flagella means composed of three parts: filament, hook, basal body. The filament is the longest portion and extends from the cell surface to the outside. Besides flagella, pili and fimbriae are also surface structure of the bacteria but do not play a role in motility. The villi are elongated tubular structure made of special protein. Fimbriae are small bristle-like fibers sprouting out of the cell. In some bacteria, they are known to help attach bacteria to rocks and streams and alter the host tissues. Ribosomes and inclusion bodies. In prokaryotes, ribosomes are associated with the plasma membrane of the cell. They are about 15 nanometer to 20 nanometer in size and are made up of two subunits, 50S and 30S units, which when present together form 70S prokaryotic ribosomes. Ribosomes are the site of protein synthesis. Several ribosomes may attach to a single mRNA and form a chain called polyribosomes and polysome. The ribosomes of a polysome translate the mRNA into proteins. Inclusion bodies. Reserve material in prokaryotic cells are stored in the cytoplasm in the form of inclusion bodies. These are not bound by any membrane system and lie free in the cytoplasm. For example, phosphate granules, cyanophycin granules, and glycogen granules. Gas vacuoles are found in blue-green algae and purple and green photosynthetic bacteria. Eukaryotic cells. The eukaryotes include all the protist, plant, animal, and fungi. In eukaryotic cell, there is an extensive compartmentalization of cytoplasm through the presence of membrane-bound organelles. Eukaryotic cells possess an organized nucleus with the nuclear envelope. In addition, eukaryotic cells have a variety of complex locomotory and cytoskeletal structure. Their genetic material is organized into chromosomes. All eukaryotic cells are not identical. Plant and animal cells are very different, as the former possess cell walls, plastid and large central vacuole which are absent in animal cell. On the other hand, animal cells have centrioles which are absent in almost all plant cells. Here is the diagram of plant cell and animal cell. Let us now look at the individual cell organelles to understand their structure and function. Cell membrane. The detailed structure of the membrane was studied only after the advent of electron microscope 1950. Meanwhile, chemical studies on the cell membrane, especially in human red blood cell, enabled the scientists to deduce the possible structure of the plasma membrane. These studies showed that the cell membrane is mainly composed of lipids and proteins. The major lipids are phospholipids that are arranged in a bilayer. Also, the lipids are arranged within the membrane with the polar head toward the outer sides and the hydrophobic tails toward the inner part. This ensures that the non-polar tail of saturated hydrocarbon is protected from the aqueous environment. In addition to phospholipids, the membrane also contains cholesterol. Later biochemical investigation clearly revealed that the cell membrane also possesses protein and carbohydrate. The ratio of protein and lipid varies considerably in different cell types. In human being, the membrane of the erythrocyte has approximate 52% protein and 42% to 40% lipid. Depending on the ease of extraction, membrane protein can be classified as integral and peripheral. Peripheral proteins lie on the surface of the membrane, while integral proteins are partially or totally buried in the membrane. These are integral proteins, and these is phospholipids bilayer. An improved model of structure of the cell membrane was proposed by Singer and Nicholson, widely accepted as fluid mosaic model. According to this, the quasi-fluid nature of lipid enables lateral movement of proteins within the overall bilayer. This ability to move within the membrane is measured as its fluidity. The fluid nature of membrane is also important from the point of view of functions like cell growth, formation of intercellular junctions, the secretion, endocytosis, cell division, etc. One of the most important functions of the plasma membrane is the transport of the molecules across it. The membrane is selectively permeable to some molecules present on either side of it. Many molecules can move briefly across the membrane without any requirement of energy, and this is called the passive transport. Neutral solutes may move across the membrane by the process of simple division along the concentration gradient. It means from higher concentration to lower concentration. Water may also move across this membrane from higher to lower concentration. Movement of water by diffusion is called osmosis. As the polar molecule cannot pass through the non-polar lipid bilayer, they require a carrier protein of the membrane to facilitate their transport across the membrane. A few ions or molecules are transported across the membrane against their concentration gradient. It means from lower to the higher concentration. Such a transport is an energy-dependent process in which ATP is utilized and is called active transport. For example, NK plus and K plus pump. Cell wall. As you may recall, a non-living rigid structure called the cell wall forms an outer covering for the plasma membrane of fungi and plants. Cell wall not only gives shape to the cell and protects the cell from mechanical damage and infection, it also helps in cell-to-cell interaction and provide barrier to undesirable macromolecules. Algae have cell wall made up of cellulose, galactans, mannans and minerals like calcium carbonates, while in other plant it consists of cellulose, hemicellulose, pectins and proteins. The cell wall of the young plant cell, the primary wall, is capable of growth which gradually diminishes as the cell matures, and secondary wall is formed on the inner side of the cell. The middle lamella is a layer mainly of calcium pectate which holds or glues of different neighboring cells together. The cell wall and the middle lamella may be traversed by plasmodesmata which connect the cytoplasm of neighboring cells. Endomembrane system. While each of the membrane is organized as distinct in terms of its structure and function, many of these are considered together as an endomembrane system because of their function. They are coordinated. The endomembrane system include endoplasmic reticulum, Golgi complex, lysosome and vacuoles. In the function of the mitochondria, chloroplast and peroxisomes are not coordinated with the above components. These are not considered as part of endomembrane system. The endoplasmic reticulum, ER. Electron microscopy studies of eukaryotic cells reveal the presence of a network of reticulum of tiny tubular structure scattered in the cytoplasm that is called endoplasmic reticulum. Hence, ER divides the intercellular space between into two distinct compartments: luminal inside ER and extraluminal cytoplasm compartments. The ER often shows ribosomes attached to their outer surface. The endoplasmic reticulum bearing ribosomes on their surface is called rough ER. In the absence of a ribosome, they appear smooth and are called SER, means smooth endoplasmic reticulum. RER is frequently observed in the cell, actively involved in protein synthesis and secretion. They are extensive and continuous with the outer membrane of the nucleus. The smooth endoplasmic reticulum is the major site for synthesis of lipid in animal cells. Lipid like steroidal hormones are synthesized in SER. Golgi apparatus. Camillo Golgi first observed densely stand reticular structure near the nucleus. These were later named Golgi bodies after him. They consist of many flat disc-shaped sacs or cisternae of 0.5 micrometer to 1 micrometer diameter. These are stacked in parallel to each other. Varied number of cisternae are present in Golgi complex. The Golgi cisternae are concentrically arranged near the nucleus with distinct convexes or the forming phase of the concave trans or the maturing phase. The cis and trans phases are organelles are entirely different but interconnected. Golgi apparatus principally forms the function of packaging material to be delivered either of the intracellular targets or secreted outside the cell. Materials to be packaged in the form of vesicles from the ER fuse with the cis phase of the Golgi apparatus and move toward the maturing phase. This explains why the Golgi apparatus remains in the close association with the endoplasmic reticulum. A number of proteins synthesized by ribosomes on the endoplasmic reticulum are modified in the cisternae of the Golgi apparatus before they are released from its transphase. Golgi apparatus is an important site of formation of the glycoproteins and glycolipids, lysosomes. These are membrane-bound vesicular structures formed by the process of packaging in the Golgi apparatus. The isolated lysosomal vesicle have been found to be very rich in almost all types of hydrolytic enzymes, hydrolysis, lipases, proteases, carbohydrates, optimally activated at the acidic pH. These enzymes are capable of digesting carbohydrate, protein, lipids, and nucleic acids. Vacuoles. A vacuole is a membrane-bound space found in the cytoplasm. It contains water, sap, excretory product, and other material not useful in the cell. The vacuole is bound by a single membrane called tonoplast. In plant cell, the vacuoles can occupy up to 90% of the volume of the cell. In plant, the tonoplast facilitates the transport of number of ions and other material against concentration gradients into the vacuole. Hence, their concentration is significantly higher in the vacuole than in the cytoplasm. In amoeba, the contractile vacuole is important for osmoregulation and excretion. In many cells, as in protist, food vacuoles are formed by engulfing of the food particles. Mitochondria, unless specifically stained, are not easily visible under microscope. The number of mitochondria per cell is variable depending on the physiological activity of the cell. In terms of shape and size also, considerable degree of variability is observed. Typically, it is sausage shaped or cylindrical, having a diameter of 0.2 to 1 micrometer and length 1 to 4.1 micrometer. Each mitochondrion is a double membrane bound structure with the outer membrane and the inner membrane dividing its lumen distinctly into two aqueous compartments. It means the outer compartment and the inner compartment. The inner compartment is filled with a dense homogeneous substance called matrix. The outer membrane forms the continuous limiting boundary of the organelle. The inner membrane forms a number of infolding called cristae toward the matrix. The cristae increases the surface area. The two membranes have their own specific enzymes associated with the mitochondrial function. Mitochondria are the sites of aerobic respiration. They produce cellular energy in the form of ATP and they are called powerhouse of the cell. The matrix also possesses single circular DNA molecule, a few RNA molecule, ribosome, 70S, and the components required for the synthesis of protein. The mitochondria divide by fission, plastids, and ribosomes and cytoskeleton, cilia and flagella, centrosome and centroids, nucleus, the main part, and then microbodies. Plastids are found in all plant cell and in euglenoids. These are easily observed under the microscope. As they are large, they bear some specific pigments, thus imparting specific colors to the plant. Based on the type of pigment, plastids can be classified into chloroplasts, chromoplasts, and leucoplasts. The chloroplasts contain chlorophyll and carotenoid pigments, which are responsible for trapping light energy essential for photosynthesis. In the chromoplast, fat-soluble carotenoid pigments like carotene, xanthophyll, and other are present. This gives a part of the plant a yellow, orange, or red color. The leucoplasts are the colorless plastids of varied shapes and sizes with the stored nutrients. Amyloplast store carbohydrate. For example, potato. Elioplast store oils and fats, whereas aleuroplast store proteins. Majority of the chloroplast of the green plant are found in mesophyll cells of the leaf. These are lens-shaped, oval, spherical, discoid, or even ribbon-like organelles having variable length and width. Their width 2 to 4 micrometer. Their number varies from 1 parcel of Chlamydomonas or green algae to 20 to 40 parcel in mesophyll. Like mitochondria, the chloroplast are also double membrane bound. Of the two, the inner chloroplast membrane is relatively less permeable. The space limited by the inner membrane of the chloroplast is called the stroma. A number of organized flattened membranous sacs are called thylakoids are present in the stroma. Flattened membranous sacs हो गया. Thylakoids are arranged in the sacs like the piles of coins called grana. On the integranal thylakoids, in addition, there are flat membranous tubules called the stromal lamellae connecting the thylakoids to different grana. The membrane of the thylakoids enclose a space called a lumen. The stroma of the chloroplast contains enzymes required for the synthesis of carbohydrates and proteins. It also contains small double-stranded circular DNA molecules and ribosomes. Chlorophyll pigments are present in thylakoids. The ribosomes of the chloroplast are smaller, 70s, and than the cytoplasmic ribosomes, 80s. Ribosomes are granular structures first observed under the electron microscope as dense particles by George Palade. They are composed of ribonucleic acid and proteins and are not surrounded by any membrane. The eukaryotic ribosomes are 80s while the prokaryotic ribosomes are 70s. Each ribosome has two subunits, larger and smaller subunit. The two subunit of 80s ribosomes are 60s and 40s while that of 70s ribosomes are 50s and 30s. Here S stand for sedimentation coefficient. It is indirectly a measure of density and size. For 70s and 80s ribosomes are composed of two subunits. Cytoskeleton, an elaborate network of filamentous proteinaceous structure consisting of microtubules, microfilaments, and intermediate filaments are present in the cytoplasm is collectively referred to as cytoskeleton. The cytoskeleton in a cell are involved in many functions such as mechanical support, motility, maintenance of the shape of the cell. Cilia and flagella. Cilium and flagella means flagellum. Are here cilia, the singular word for cilia is cilium, and flagella for singular word of flagella is flagellum. Are here like outgrowths of the cell membrane. Cilia are small structure which work like oars, causing the movement of either the cell or surrounding fluid. Flagella are comparatively longer and responsible for cell movement. The prokaryotic bacteria also possess flagella, but these are structurally different from that of eukaryotic flagella. The electron microscope study of the cilium or flagellum so that they are covered with plasma membrane. Their core is called the axoneme. Possesses a number of microtubules running parallel to the long axis. The axoneme usually has 9 doublets of radially arranged peripheral microtubules and a pair of centrally located microtubules. Such an arrangement of axonemal microtubules is referred to as 9+2 array. The central tubules are connected by bridge and is also enclosed by a central sheath which is connected to the one of the tubules of each peripheral doublets by a radial spoke. Thus, there are 9 radial spokes. The peripheral doublets are also interconnected by linkers. Both the cilium and flagellum emerge from centriole-like structure called the basal bodies. Centrosomes and centrioles. Centrosome is an organelle usually containing two cylindrical structures called centrioles. They are surrounded by amorphous pericentriolar materials. Both the centrioles in a centrosome lie perpendicular to each other, in which each has organization like cartwheel. They are made up of 9 evenly spaced peripheral fibrils of tubulin protein. Each of peripheral fibril is a triplet. The adjacent triplet are also linked. The central part of the proximal region of the centriole is also proteinaceous and called the hub, which is connected with the tubules of the peripheral triplets by radial spokes made up of protein. The centriole form the basal body of cilia or flagella and spindle fiber that give rise to spindle apparatus during cell division in animal cells. Nucleus. Nucleus, as a cell organ, was first described by Robert Brown as early as 1831. Later, the material of the nucleus stained by basic dyes were given name to chromatin by Fleming. The interphase nucleus of a cell when it is not dividing, has highly extended and elaborate nucleoprotein fibers called chromatin. Nuclear matrix and one or more spherical bodies called nucleoli. Electron microscopy has revealed that the nuclear envelope, which consists of two parallel membranes with a space between 10 to 15 nanometer, called the perinuclear space, from the barrier between the material present inside the nucleus and that of cytoplasm. The outer membrane usually remains continuous with the endoplasmic reticulum and also be a ribosomes on it. At a number of places, the nuclear envelope is interrupted by minute pores which are formed by the fusion of its two membranes. These nuclear pores are the passages through which movement of RNA and protein molecules takes place in both direction between the nucleus and the cytoplasm. Normally, there is only one nucleus per cell. Variation in the number of nuclei are also frequently observed. Can you recollect names of organisms that have more than one nucleus per cell? Some mature cells even lack nucleus. For example, erythrocytes of many mammals and sieve tube cells of vascular plant. Would you consider these cells as living? The nuclear matrix of the nucleoplasm contain nucleolus and chromatin. The nucleoli are a special structure present in the nucleoplasm. The content of nucleolus is continuous with the rest of the nucleoplasm, as it not a membrane-bound structure. It is site for active ribosomal RNA synthesis. Larger and more numerous nuclei are present in cell, actively carrying out protein synthesis. You may recall that the interphase nucleus has a loose and indistinct network of nucleoprotein fibers called chromatin, but during a different stage of cell division, cells so structured chromosomes in place of the nucleus. Chromatin contain DNA and some basic proteins called histones. Some non-histone proteins are also RNA. A single human cell has approximately 2-meter long thread of DNA distributed amongst its 46 pairs of chromosomes. 46 chromosomes and 23 pairs of chromosomes. You will study the details of DNA packaging in the form of chromosome in class 12. Every chromosome, usually only in dividing cell, essential has a primary construction or the centromere, on the sides of which disc-shaped structure called kinetochores are present. Centromere holds two chromatids of a chromosome based on the position of the centrosome. The centrosomes can be classified into four types. The metacentric chromosome has middle centromere forming two equal arms of the chromosome. The submetacentric chromosome has centromere slightly away from the middle of the chromosome, resulting into one shorter arm and one longer arm. In case of acrocentric X chromosome, the centromere is situated close to it and forming one extremely short and one very long arm, whereas the telocentric chromosome has a terminal centromere. Sometimes a few chromosomes have non-staining secondary constrictions at a constant location. This gives the appearance of a small fragment called the satellite. Microbodies. Many members bound minute vesicle called microbodies that contain various enzymes are present in both plants and animal cells. So our chapter is finished. Thanks.